Hey guys, so this is going to be our very first lecture video. I just record them like it would be in the classroom, like I'm just talking at you. Obviously, I don't get a whole lot of interaction. So to alleviate that, I want y'all to please watch these videos like I'm talking at you in the classroom, and then hit me up with questions. Email me. We can do face-to-face Zooms if we want as a class. We can all come together at a time that we want to meet at during office hours, and that can be our designated time for class that we can meet and talk about the different chapters and the information and the questions you might have. Let me know, and we can vote on this, try and do this. But definitely individually, questions are okay. Watch these things. I will tell you, tips and definitely things to know for sure. So please watch these videos. I think they're helpful. I hope you think they're helpful. Let's get started on the different chapters. This is chapter 1. There are 26 chapters in this micro book. 26. We are going to go through 24 of the 26 chapters. So this is the very first one. We're going to do at least a chapter, a normal lecture period in this class. So chapter 1 is discovering micro. What is the invisible world? So let's keep going. Microbes are going to be, microorganisms are going to be the little bitty guys. These teeny tiny guys, usually they are things that we cannot see with the naked eye. So usually we're talking about bacteria, viruses, stuff that we don't normally see. There are going to be other things that will cause infections that are things that we can see with the naked eye, like parasites. So most of the time when you think of micro means small. So you're going to think of the little guys. But it can mean things that you can see with the naked eye too. Helminths fall into that category. Most of these guys are harmless. You watching this class right now, me, we are covered in micro. We are covered. We have our own biomes, and different parts of your body are going to have different biomes on there. You're going to have bacteria. You are going to have fungi inside your body, outside your body. Most of these guys, again, are harmless, and they do good for us. They help us break down our food. They help us with our vitamin absorption. They help us defend against the bad guys. So most of these guys are harmless. They are good. We are covered in them. But some of these other guys are pathogenic. They're bad guys. So we're going to talk about all of these guys in this class. They are going to play a fundamental role in our ecosystems. Yeah, you're going to find these guys everywhere. I can't remember where in the lecture it is in this chapter where we talk about how old the Earth is, and it's 4.6 billion years old, by the way. And when we divided up when we get into taxonomy, there were 3 domains, and 2 of the domains are prokaryotes and 1 of them are eukaryotes. The prokaryotes came first. Bacteria and archaea came first, and they are everywhere because they've had all the time to go and evolve into all the different places. So these guys are everywhere. They are the base organisms no matter where you go. Arctic poles, hot springs in Yellowstone, these guys are here. So they are going to be, the backbone of the food webs. And we still don't know everything about everything. Nobody knows everything about everything. We're still learning. That is so true right now, today, in the middle of a pandemic. So we are still so much left to learn. We can see this picture of coming from Deepwater Horizon of an oil spill and an organism, a sea turtle, covered in oil. And we can talk about remediation. Bioremediation is being able to use an organism to remediate something. So there are organisms out there, like this guy, that can help break down those fuels, those oils that have been released. They can actually use those and metabolize them and break them down. So we can bioremediate when we know about microorganisms. So that's something super cool that we can do with them. All right, let's go to this first section of chapter 1 in this video because I don't want to make the video super duper long. I like to chop them up into the different sections. So we're going to talk about what the ancestors knew. So before we get there though, the textbook does these things called case studies. Every chapter I think has one. In this case, our case study that we start out with in chapter 1, her name is Cora. She's 41, so she is 1 year younger than me. 41-year-old female, and her signs when she comes in. So what's her signs and symptoms? She's got a fever, so symptoms are fever and headache. Well, that's pretty standard, and that could be almost anything. She's getting confused, and she's got a stiff neck. Stiff neck series. With all of these together and the stiff neck, we're getting worried. So differential diagnosis, we're thinking meningitis. So something's in her spinal fluid, something's going around in her CSF. What is it? What could cause these symptoms? What do we think it is? So what are possible causes? Bacteria. Is it bacteria or is it viral? Which one would you rather have if you had meningitis, bacteria or viral? We'll get to that in a little bit. Later on this semester, we're going to talk about that and the difference between bacterial and viral and how fast one acts. But again, it could be bacterial, it could be viral, it could be fungal. It could be fungal causing meningitis. It could be heavy metals or medications that are making her react this way with these symptoms. So if we're thinking meningitis, we want to do a spinal tap, test the CSF, see what's going on in there. And maybe we can figure out in the CSF, is something growing in there? What is it? And now we can treat it once we can diagnose what's in there. So this is the very beginning, trying to treat Cora, figuring out what's going on with her, coming up with her differential diagnosis, and then we'll track Cora throughout chapter 1 and figure out what's going on with her. All right, so we're going to pull you back into the normal chapter 1 content. So coming back in, we know that people have been using microorganisms since we started recording history. Fermented foods are where it's at for millennia. If you enjoy a drink, an alcoholic beverage, let's say wine, beer, these things are fermented. They are fermented. If anybody has tested their craftiness and trying to do homebrew on any of this stuff, you know that fermenting is a process. And so what it is when the organism takes the carbs, it takes the sugars, and it breaks it down, and it produces the alcohol. It will ferment the substance. If you enjoy, it doesn't have to be beer or alcohol. You could be non-alcoholic and you're still using fermented foods. If you enjoy something as simple as bread, I love me some carbs. I'm always going to talk about eating, by the way, just so you know. I'm always thinking about it all the time. So I love bread. And if you love bread as much as I love bread, you like the bread that is fluffy versus unleavened bread. Like if, I don't know how many people are Christian or Methodist or Catholic, but those wafers, God, my religion background is lacking, because I know there's other besides Catholic. I'm Catholic and it comes to my mind. But if you get those unleavened wafers, those aren't risen. They haven't risen. What causes the bread to rise? It's the microorganisms. So in this case, it's yeast. Yeast are little eukaryotes, and they undergo fermentation. And then those little gas bubbles come up as they ferment, and that makes the dough rise. So if you enjoy bread, like I enjoy bread, you're enjoying a fermented object, a fermented food. If you love cheese, I'm also a big fan of cheese late at night. That's my go-to snack, is cheesy things. Salty is my thing. Cheese and chocolate. Cheese, if you like cheese, non-pasteurized cheeses, or even the pasteurized ones that have had the organisms heat killed out of those, that's a fermented food. Yogurt, kefir, kombucha. If you pick a container of some of that up at the local grocery store or natural food market, that stuff's pretty good. All of this is fermented food. If you like sauerkraut, I could go on and on. We're going to move on. Fermented foods have been around forever, and we have been using them for a long time. So I got the first lecture quiz question here. If we were face-to-face, we would all go into our lecture quiz tool and we would start to answer these questions. Since we are not face-to-face, y'all will log on to D2L this week and you will take care of the lecture 1 chapter quiz, and it's 5 questions based off of the material found in the textbook, found in the chapter outlines that I'm telling you right now in lecture. So hopefully everybody can use that material and look through it, review it, and then go in and take that lecture quiz and see how you do. Lecture quizzes are designed to be, they're not super easy points, they're not gimme points, but you should be able to figure out what the proper answer is. I'm not making them hard. I'm not trying to trick you with these. These are supposed to be questions that you should be like, oh yeah, yeah, yeah. We just talked about that in the video and I remember what that answer is. So that's our first lecture quiz question. You're gonna have 5 for every chapter. Okay, so more proof that we have been using microorganisms for a long, long time. This one's super cool. It is the Iceman, Otzi, or Otzi. I'm probably mispronouncing that. He was found in the Swiss Alps. He is over 5,000 years old, 5,300 years old. And he was well preserved, so well preserved that we could tell what microorganisms were inside of him. And this person, Otzi the Iceman, he was infected with a couple of different microorganisms. So he has trichuris, he's got whipworm, so he's got helmets, he's got worms in his gut. And he also has Lyme disease caused by Borrelia burgdorferi. So he's got Lyme disease and he's got whipworms in his belly. So, okay, so he's sick. What does that tell us about using microorganisms? Well, they figured out that he was also, when they went through his clothes and his items on his person, they found that he had these woody fruits on him and that there was a fungus that lived in this fruit and that the fungus had antibiotic properties and it was also a laxative. So if you have a belly full of worms, if you're eating something that's a laxative, that's going to help you try and pass those out. So he's trying to treat himself. He's trying to self-medicate using that fruit with the fungus on it. So that's super cool that we know that they were doing this for sure over 5,000 years ago, that people were trying to treat themselves with what they knew at the time was working. So antibiotic properties to try and combat the infection, combat the Lyme disease, laxatives to try and get rid of those worms in his belly. He also had tattoos. And so they think that these tattoos were made, so they sliced the skin and then they put herbs in there and then they burnt the herbs. So he had tattoos all over and they think that this was also another form of treatment as these herbs were put into his person, into his skin. So another example of people using microorganisms a really long time ago. So moving on from Otzi, we are going to talk about historical names. There's a few chapters in here where we do talk about names. You're going to need to know who did what and what is their famous nickname and what were they important for? What was their big discovery? So we first started thinking about things like this in before Christ in ancient Greece. We mentioned, speaking of Christ, in the Bible, they mentioned leprosy, which we now call Hansen's disease. But they mentioned quarantining people, right? We quarantine the sick, and we are currently doing that today, right now. But this is not a new concept. Again, pre-biblical, Old Testament times. Ancient Greek. The ancient Greeks believed that it was something in the air, and it was bad air. So mal means bad, malaria, malaria. The bad air, it caused this miasmic, this bad air is the source of the illness. So if there's bad air around, miasmic air around, then that is why somebody is getting sick. So if we can stop that and improve our hygiene, then people should get better and not as sick as they used to. So the Romans took care of that. The Romans were some of the ones that we have proof of that they tried to improve hygiene. So instead of everyone just, going wherever and just leaving, feces out there, they tried to take care of it. So what they did is they built sewers. Their biggest one is called the Cloaca Maxima, and you can actually still see it. Not that anybody wants to go and see a sewer. That's gross, but it actually is still there. Like the pieces of it are still there, and it still empties into the river Tiber. Of course, it's not emptying out fresh stuff into the river now, but you can still see where it empties out into the river. Nothing's coming out of it though. So they built a sewer to get rid of the feces. We have this lovely latrine that is designed so everyone goes in one place, and then we are going to need to flush it away. Let's get rid of that stuff. So how do we flush it away? We're going to use aqueducts. And so Romans are known for their aqueducts. So they were able to take the fresh glacier-fed spring snow melt water from the mountains and pipe that stuff into the city using these aqueducts. And it's super cool architecture. It is still around and it is still being used. You can enjoy fresh mountain spring water in the city of Rome right now, today, from these aqueducts bringing water in. There's multiple fountains in the city, all over Rome, where you just literally just go up to the fountain and you put your water bottle, your reusable bottle there, and you can drink that water. It's safe. So they were bringing in fresh water, and that was helping to give everyone fresh, clean, healthy water that's not, questionable, sitting there growing things. This is fresh flowing water that everyone's able to drink. And then they're also going to use that water to flush away all the feces from these latrines. So hygiene made a huge impact in Rome. And we can still see this stuff today. You can still go and enjoy that fresh water today. It's really neat. They have an app out there that shows you where all these little things are. They're sprinkled again all over Rome. It's really cool. It's really cool, especially when it's hot like it is right now. I have a travel bug. I have wanderlust, and I like to travel. And I encourage everyone to do so. And obviously, we cannot do it now. Don't go now. But when things get better, go see, check out these places. They're super cool. And this is something that we're talking about now in class, but you can still see it today. If things were better. So go and travel, see things, go places, do things. All right, other names that we need to know. So the Greeks, bad air, miasma. Romans are trying to take care of that bad air. If we can flush away all the poo and we're not sitting there, if it's not sitting there festering, we're going to clean that away with sewer systems. That's going to improve everybody's health. So now we moved on to some initial Greek physicians, some more Greeks that you're going to need to know. So Hippocrates is definitely a name that you should know. Anybody in the health field should recognize this name. The Hippocratic Oath, the do no harm, that's Hippocrates. So he is the father of Western medicine. That's Hippocrates. And so he was the first guy out there that was telling his colleagues and other people in the field, it's not supernatural. It's not, God or gods or whatever, ghosts, whatever you want to think. It's not supernatural. There's some natural cause for this. It's got to be the patient or what this patient is exposed to around them that is causing the patient to be ill. So he's the father of Western medicine. This is Hippocrates. Know him and his moniker, what he did. All right, so now we're going to move on to another name. So we have Thucydides, and so he is the father of scientific history. So he looked at cause and effect. How did he look at cause and effect? Well, we are not the first plague. I'm sure we all know this. Everyone has probably heard of the Black Plague, in the Middle Ages with the bubonic plague. Well, there was another plague even before that, and that was the Athenian plague in Athens, in Greece, and it took out a third of the population. And so what Thucydides discovered was that the people that were affected by the plague, got the plague, and survived, they didn't get it again. So he was observing these cause and effect relationships using scientific bases, hypotheses, and studying it with these observations. So he is the father of scientific history based on his observations that were happening during the plague. during the Athenian plague. All right, so Marcus Terentius Varro, he was a writer, he was an author, and he thought that he kind of, pulled off of the belief that, we've already said that it's not supernatural. Hippocrates said it's not supernatural. We've talked about this bad air. So it's got to be something small. So Marcus Varro, he thought that it's got to be something small. Maybe it's something so small that we can't see that's causing the disease. So he was thinking that things were, possibly floating in the air and then getting into the body, like we said. So he is one of the other original scientific Greek people names that we need to know. Moving on, so we got some more names. So Anthony Leeuwenhoek. Anthony Leeuwenhoek. Okay, so we said these Greek guys, they were leaning towards it's not supernatural and there's tiny things out there. We think there's tiny things out there, but we can't prove it. By the time Leeuwenhoek comes along, so he's 1600s, 1700s. By the time he comes along, guess what we've discovered? Guess what we have invented? We have invented microscopes. So we have the first microscopes going on right now. And Leeuwenhoek grabs some dirty old pond water and puts it under his microscope and looks, and lo and behold, he sees these little things, wee little beasties, or animalcules. I think wee little beasties is way cuter. But animalcules is what he called them. So he was probably looking at protists or maybe some rotifers, bacteria, but he was looking at creatures. So now we have proof that in things that we couldn't see before, now we can see them with a microscope. And he called them animalcules or wee little beasties. This was Leeuwenhoek. Leeuwenhoek, definitely going to ask you about Leeuwenhoek. I'm definitely going to ask you about these next two guys too. So in the golden age of microscopy and microbiology, we have two names that stand out, and we will talk about them again later on. The first is Pasteur. If we were talking about, remember we were talking about cheeses before and pasteurized cheeses? We have Pasteur to thank for that. So he came up with the process that we're going to talk about later on of showing things that life did not just occur. Spontaneous generation does not happen. We can sterilize things and keep them that way. We can pasteurize things. So that's what he did. That is what he's famous for, is pasteurization. Well, later on, we'll go into details of how he did it with this little swan neck flask, a specialized little glass. But that's Louis Pasteur, pasteurization. And then I think this is the last guy of this video, is Robert Koch, Koch's postulates. We will talk about Pasteur and Koch in the future. But Koch was really, really cool. He used his postulates to be able to isolate things. So he figured out what the cause of some things are. He figured out what causes anthrax and cholera and tuberculosis. He figured out these guys. He found the bacteria that caused them using what we now call Koch's postulates. So later on this chapter, not in this chapter, later on, we will go through Koch's postulates and how he did what he did. Same with Pasteur. But right now, know their names, have a general idea of what these guys are famous for. Tools. It makes me sad that we don't have a face-to-face lab for y'all right now. If we were face-to-face, we'd be able to go through all of these. Right now, at the time that I'm making this video, we are not. We are fully online. But obviously, microscopes are needed for microbiology lab. We are going to talk about stains and dyes, and you'll be able to see pictures of them, and we will definitely use all of these virtually in Labster, because you'll be able to run the experiments in Labster and see what that is. But we're going to use stains and dyes. Those things are going to help us differentiate between bacteria, because some things will absorb stains and some won't, and some will absorb different stains but not other stains. So they'll help us differentiate between different types of things. There's also different growth media out there. We use agarose in our petri dishes a lot, but you can also add things to the agarose. We may use sheep's blood. We may use other things in that. So there's different types of media in the petri dish. There's different types of media in the tubes that we put in there. Some tubes will, when we get into selective and differential media, we'll talk more about that. But there's different types of media in the test tubes. When you need to transfer organisms in this class, it's doing it virtually, but you're really doing the same stuff that we're doing face to face. You're just doing it in Labster. So you will transfer organisms from one tube to an empty tube, your sterile tube that's got nutrient media in there. And you will, if I'm gonna pretend like my two fingers are the tubes. So you would take your loop and you put it on your baccinerator, micro incinerator. Ours are called baccinerators. And so you'd heat up your loop and you'd sterilize it. And then you're going to have your stock, which is going to have your organism in it. And you dip it in there. And now you got your organism on there. And then you are going to put it into the tube that doesn't have anything in there. And then we're going to cover them up. And we will put them in the incubator. And then we're going to grow them, hopefully, if everything works right. That's what should happen. So you will be able to play around with these virtually and use all of these tools virtually. Here's a great picture of the loop, and there are going to be steps involved on making something like this. So we have a streak plate. This is a streak plate. And so there's different steps, but what you do is you take your mystery organism that you're not quite sure what you might have. So you take that loop and you sterilize it. So it's totally sterile, nothing's living on there. You take a scoop. I don't know if y'all can hear my dog. She's being really weird in the background. I have 2 dogs, so you might hear them. And I have 3 kids, and you might hear them too right now. Just the one dog, and she's being weird. Anyway, you take your loop, baccinerator, heat it up, sterilize it, let it cool, because you don't want to fry your organisms. You take a scoop of your mystery organism, and then you're going to put that scoop onto a fresh petri dish that has nothing in it, and then you're going to swipe, 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 and lay down this stuff. So that would be this section over here, section number 1. And then you're going to take that loop, you're going to sterilize it, and then you're going to come back after it's cool, let it cool, and then you swipe, swipe, swipe again. And then we're over here in section 2, because you're going to go from the new stuff, from the empty media, into the bacteria, and you shake it, shake it, shake it again. Then you're going to sterilize it and let it cool and come back and do that again over here. And you kind of drag it out. And so what you're doing every time that you are sterilizing and then going back in, you're thinning down your bacteria. So as you go all the way to area 3, you will have individual colonies. And this is what is needed to study the organism. Get those individual colonies. What color are they? What's the growth pattern? What does it look like? And then you can sample those, put them on a slide, because you have isolated them with this streak plate. So these are all different tools that you will talk about, you use in your lab, hopefully virtually in Labster. All right, so that is the end of this first video. I have talked way too long for this first video, but let me know if you have questions, and we will do more videos on the rest of the chapter.